Thank you, Todd. I appreciate the introduction and also thank you for the invitation. I'm honored to be the first woman to present this lectureship. And tonight I'm going to talk about how to exploit opportunities to create disruptive change within your industry. So just to begin with, what is disruptive change? Well, disruptive change occurs because of an innovation in either a business model, a technological advance, or perhaps a superior alternative product in an already mature industry. What I hope to provide to you tonight is some information about how I've exploited opportunities to make disruptive change in the two markets for the two companies that I've had. So the ways that we can exploit opportunities include the use of new technologies to replace existing methods, or implementing a new business model, using customer feedback to improve products or services, expanding your customer base, and or creating customer loyalty. And so as I go through my lecture, hopefully you'll identify some of these opportunities that I've outlined here. So just briefly, my career path is I'm from the Pacific Northwest. I got a biology degree at Washington State University, and then I went to the Medical College of Virginia for my PhD. From there, my first job was at Baylor College of Medicine in Houston, Texas, and I was there for almost 12 years. Then I moved back to Spokane, Washington, and I was at Washington State University on the Spokane campus for a number of years. And I was also a clinical cytogeneticist at Sacred Heart Medical Center. But if you think about when you start your career, when does your career really start? And, you know, as far as back as you can remember, people have probably been asking you, what do you want to be when you grow up? And for those of you who are currently college students watching this lecture, people are probably still asking you that. What do you want to be when you grow up? And I knew what I wanted to be since I was a sophomore in high school, which is really unusual. But when I was a sophomore in high school, in my biology book, there was one chapter on in the biology book, there's one chapter on genetics. And in that chapter, it talked about Down syndrome. And this is a little girl with Down syndrome. And Down syndrome is caused by having an extra chromosome 21. And I found this so fascinating because I always thought more was better. So why having an extra chromosome? Why would that cause some of the difference in facial appearance that you see here in this little girl? It can cause heart abnormalities and other medical problems. And I decided this is what I was going to do. I was going to devote my life to studying Down syndrome. And so I decided I wanted to be a cytogeneticist. Well, as a sophomore in high school, I didn't know the word cytogeneticist. I didn't know that existed, and I didn't know how to get there, but I knew that I wanted to study chromosomes. So cytogenetics is the study of chromosomes. Humans have 46 chromosomes or 23 pairs, and they're shown here. Of course, they don't look like this under the microscope, but we pair them up so that we can see each of the pairs of chromosomes. Aneuploidy is extra or missing chromosomes. So this is Down syndrome. So here's a karyotype of an individual with Down syndrome. You can see there's 3 copies of chromosome 21 when there should only be 2. Now, during my dissertation, when I was at the Medical College of Virginia, I studied a rare form of Down syndrome where the extra chromosome 21 is actually attached to another chromosome. And it's most commonly attached to a chromosome 14, as you can see here. It's sitting up here at the top. What I found out during my dissertation research, when I got my PhD, is that this 21, when it attaches to a chromosome 14, when it was new, when it occurs new, it always happens from the mom. So this was something that wasn't known. And it's very interesting because if you think about it, little girls are born with all of the eggs they will ever have in their lives. So when these two chromosomes, this 14 and this 21, became attached, they actually became attached when the mom was a fetus, when her eggs were being produced. So you guys can think about that for a minute. That's a little bit of a brain twister. So most of the time in clinical genetics, which are a collection of clinical features or medical features found in individuals, they're usually identified in what we called a phenotype-first approach. And once there was a group of patients that all seemed to have the same clinical features, then researchers would try to investigate the genetic basis of those syndromes. So for example, in Down syndrome, it was discovered that it was caused by an extra chromosome 21. But as techniques improved, more chromosome abnormalities were identified, and this allowed for what we called a genotype-first approach, where we would find a group of patients based on having the same genetic change, and then we would then look at those individuals and see what features they had in common, and then that would constitute a new syndrome. So this is what I did when I was at Baylor College of Medicine, and the first syndrome I worked on was Monosomy 1p36. Now, Monosomy 1p36 is a deletion of the very tip of the short arm of chromosome 1. And there's an arrow here because without that arrow, it's very difficult to see. We would use techniques. So over here in this picture with the blue chromosomes, we would use techniques such as fluorescence in situ hybridization, where we could see a deleted chromosome 1 as compared to a normal one. So a normal one has the green probe, which indicates a chromosome 1, and then the red probe indicates the end of the chromosome, and you can see on this one it's missing. When we found 14 cases in the laboratory that I worked at Baylor that had deletion of chromosome, the short arm of chromosome 1. And when we looked at the patients, we found that they were very similar in that, again, they had very similar facial features. They had little pointy chins, small mouth, very flat face, and typically had very straight eyebrows. Well, when I first presented this at a genetics conference, a national genetics conference, I was literally called crazy by people because we found 14 cases and no one in the audience, and there was probably 2,500 people in the audience, had ever seen a case of this. They thought I was seeing things. Well, we started looking through the archives and over the years, and in Houston alone, we found that 1 in 5,000 newborns had a deletion of 1p36. It turns out it's actually one of the most common chromosomal syndromes in humans, and it was simply being missed because we were looking through a microscope, and it was very easy to not see this little light fluff at the end. So in my research lab, we decided to map these breakpoints, because not all the patients had the same breakpoints. So shown here is just a map of the most distal region of the short arm of chromosome 1, and every one of these yellow bars indicates a little piece of DNA. And we would use these little pieces of DNA to walk along the length of the chromosome to find where the deletions were. So if this piece right here was retained, then and this piece was missing, then that means the breakpoint was between these two little pieces of DNA. An experiment like this in one patient would take us about 18 months. Well, I went to the American Society of Human Genetics meeting, and this was probably around 1997 or so, 1998, and saw a presentation about a microarray. Well, a microarray turned out it was actually something pretty simple, where you take all these pieces of DNA that you know about, and you put them on some sort of solid support like a glass microscope slide. And then this is just a picture of all those spotted arrayed clones on the glass microscope slide. So we came back from that meeting, we were very excited to try to create our own microarray for the distal short arm of chromosome 1. And we did that. And this is actually the very first experiment using one of the arrays that we spotted ourselves. And we could quickly see that all this region down here was chromosome loss. So this was loss of a part of the short arm of chromosome 1. And this red bar indicates normal copy number. So this is the normal chromosome 1. And so we could very quickly see that the deletion was between a clone here and a clone here. That was where the breakpoint was. And then we would set out then to clone the breakpoints because we wanted to understand why was chromosome 1 breaking in this region. This experiment literally changed my life. We went from testing patients, their DNA, taking about 18 months per patient to map a breakpoint, to an overnight experiment. And so I went to the chairman of the department at Baylor, and I said, I want to spot the entire genome on a microscope slide, and I want to use this to do to test for chromosome abnormalities rather than looking through a microscope, because I know that when I look through a microscope, I'm missing things. I'm missing things too small to see. So when I went to my chairman, I said, there's advantages to using microarrays for cytogenetics. First of all, the cells didn't need to be cultured for 3 days. We didn't have to wait 72 hours to even prepare the chromosomes. We could do genome-wide testing, and it can be accomplished in 24 hours. All previously known gains and losses that you could see through the microscope could be identified. Deletions and other abnormalities too small to see through a microscope should be identified. And it allowed for us for this genotype-first approach to identify new syndromes. Well, the chairman said no, and he had reasons for saying no. And so these are some of the first obstacles that I had to overcome. So first of all, he told me that microarrays at the time were research use only, and they were not to be used for clinical use, and therefore I could not use them in my cytogenetics laboratory to make clinical diagnoses. Secondly, there were big companies such as Affymetrix and Roche that had the intellectual property rights. And thirdly, he was concerned that we might identify something smaller than what you could see through the microscope, and how would you possibly interpret that DNA change? So I left. I left Houston, I left Baylor. I was a tenured full professor, and I moved back to Spokane, Washington. I moved back to Spokane, Washington with the help of Harry Mielke, who was the chancellor of WSU Spokane at the time, and Skip Davis, who was the CEO of Sacred Heart Medical Center at the time. And I started Signature Genomic Laboratories. So Signature was started here in Spokane, and it was a joint venture between Sacred Heart Medical Center, and again, Skip Davis, and Pathology Associates Medical Laboratory, or PAML, and Tom Tiffany was the CEO of PAML at the time. And so they, with my scientists, we started together Signature Genomics. So Signature Genomics' vision was to improve the detection of cytogenetic abnormalities among those with intellectual, developmental, and physical disabilities. And what's so interesting is I never set out, the first slide I showed you, what are you going to be when you grow up? And I said, cytogeneticist. I never set out to be a business person. I never set out to start my own company, but it was what I needed to do to get this done, because I knew that this was better than looking through a microscope. And Signature Genomics was the first cytogenetics laboratory in the world here in Spokane, Washington, to use clinical diagnostic microarrays for the detection of chromosome abnormalities in children with developmental disabilities. So what we really did was we transformed this chromosome analysis, labor intensive, through the microscope. You needed to have expert eyes in order to see these little tiny changes in these banded chromosomes. We really changed it to an information-based discipline where we had computer technology where we could quickly see the deletion. So here's the deletion of the short arm of chromosome 1. Our software that we developed would flag the abnormalities and would report out the abnormalities. So again, from having to culture cells, which took 72 hours, within 24 hours, we could identify abnormalities. Well, Signature Genomics was really a fairy tale. So we started in the summer of 2003, and we launched our first product in March of 2004. And by September of 2004, we were profitable, and we were profitable every day after that. So it was really quite incredible. And of course, if you're starting a company, this is the kind of chart you want to see, right? It's growing every year. So it's a beautiful chart. And we had a lot of recognition. So we were named to the Inc. 500. We were one of the fastest growing private companies in 2008. We were ranked 114 in the nation. We were 5th in all of the companies in healthcare, and we were the 4th fastest growing company in the state of Washington. In 2004, we had 6 employees, and by the time we entered for the Inc. 500, we had 65 employees. The company went on to be recognized in 2009 and 2010 as well. By 2010, we had 121 employees, and this was the year that Signature Genomics was acquired. Now, Signature continued to get criticism. So I had those criticisms when I was at Baylor, but when we would go and present at scientific conferences, we would continue to get criticism. So for example, the identification of DNA changes of unknown clinical significance. So now we're finding small changes in the genome, and we are being criticized. Well, what does that mean? Well, what did we do about it? Well, we curated a large database to track these DNA changes and outcomes. So we would go back to the clinicians that submitted samples to us and ask them about their patients, ask them about the medical problems that these patients were having. And we published these frequently in the medical literature so that others could learn from them. We were also criticized that the techniques were not easily integrated into other laboratories. So this is probably a really, really bad business model, but we traveled to other labs and we actually taught them how to do microarrays. And so we created our own competition. But we also had yearly signature conferences held at the Doubletree here in Spokane. And we pivoted. We started selling signature chips because if laboratories were going to do this testing themselves, and they were going to do this testing themselves because we were teaching them, then perhaps we should sell them arrays and have another stream of revenue. So we sold signature chips. And we also provided our genoglyphic software tools. We were also criticized that we were not sharing our data. So we had thousands and thousands of cases in our database, the largest number of cases in the world. So we started providing access to the large database through genoglyphics. We actually had a public access, but it was limited. It only showed certain amounts of data. And then we had a subscription access to the entire database for those who needed genoglyphics to interpret their microarrays and who were buying signature chips from us. We made significant contributions to the medical literature. So we published more than 45 different papers from 2004 to 2010, and we identified more than 12 new genetic syndromes. So I was looking back through my CV, and I actually stopped counting at 12. So I'm very proud of all the new genetic syndromes that we identified, because this provides answers to families who are searching for diagnoses and for reasons for their children's medical problems. We improved the diagnosis of cytogenetic abnormalities from about 3 to 4%, if you look through the microscope, to 20 to 25% using microarrays. And these are in children with developmental disabilities. And we published the first and the largest series of postnatal and prenatal diagnoses using microarrays. So what does molecular cytogenetics look like today? Well, first of all, the microarray testing is endorsed by the American College of Medical Genetics. It was endorsed in 2007, really in the middle of Signature's rise. And so this was really important to the success of Signature. Arrays are now the first test done in a child with developmental, physical, or intellectual disability. And all cytogenetics laboratories in the world either have access to microarrays or they perform the testing in-house themselves. So as I said, Signature Genomics was acquired in 2010. And by 2012, I was looking for something different to do. So I still wanted to do something in genetics, but I really wasn't sure what I wanted to do. And I knew I wasn't ready to retire yet. And a friend of the family had just had a litter of chocolate Labrador retriever pups. So I was over at her house and we were talking, I was playing with the puppies. And she said, do you know how hard it is to get genetic testing done on these puppies? And I said, no, tell me about it. Because I didn't know anything about genetic testing in dogs. I didn't even know there was genetic testing in dogs. And she would do genetic testing in her dam and her sire. And she would also do genetic testing in the pups if she needed to see if one of them was a carrier. And she was having to send samples to multiple different laboratories and even had to send samples to a laboratory in France because they were the only ones that did a particular DNA test. And I thought, well, somebody ought to be able to do better than that. Why isn't there one-stop shopping? Why isn't there a lab that just can do everything? Why would you have to send your sample to multiple different laboratories? So I decided to start Paw Print Genetics. So we did a lot of market research to determine whether or not there was, because this was a mature market. There were already laboratories that had been out there for 20 or 25 years doing genetic testing. So was there something that we could do differently that would attract people to use our company? So our vision was to raise the standard in canine genetic diagnostic testing, carrier screening, and customer support. And we believed that if we educated the clients in the community on the importance of optimal canine genetic health, that they would then use genetic testing. And Paw Print Genetics was actually the first canine laboratory to use high throughput genome technologies for fast, efficient screening. And when we came on the market in 2013 with our tests, our menu had 86 tests. Most laboratories were offering 10. So we immediately had the largest menu of tests available. So when we were doing our market research, what I did was I took a cheek swab from Trixie the wiener dog, who was our dachshund at the time, and I sent samples to all of the known laboratories in the United States so I could understand what were they testing, what was it like to place an order, what was it like to receive their results, did they send me a kit or did I have to find my own kit? Then I had to look at the whole industry. Was canine genomics really a big enough industry for us to get involved? So first of all, selective breeding in dogs and artificial selection has produced all these different kinds of dogs. So anything from a Chihuahua to a Great Dane. And the selective breeding of these dogs have not only fixed the different DNA changes, the variations that cause a Chihuahua to look like a Chihuahua and cause a Great Dane to look like a Great Dane, but it also, unfortunately, has fixed certain disease mutations in these populations. And there's now more than 320 known mutations that cause medical problems in dogs. In the US alone, people spend over $100 billion annually on their pets. So it certainly is, genetic testing is a small part of this, but it seemed like it was a big enough market to have another player. The canine genetic testing market was underserved. And this is what I identified through our market research. There were a few companies out there at the time. I think there was 8 companies in the US, but each was doing a handful of tests. They were performing poor to mediocre customer service and reporting. So I got reports back from Trixie the wiener dog. And in fact, one of the companies just sent me an email and all it said was carrier. And I couldn't even remember what test I had ordered from them. So carrier of what? And then I decided to start talking to customers like my friend who had the litter of chocolate labs. And they described to me that they would get their reports back, but they weren't really always sure how to use the information. So I felt that based on this information that there probably was room for another company. And we thought that if we provided better service and education, that breeders and dog owners would be likely to use testing prior to breeding or buying. So how did we disrupt a mature market? Well, we did it by filling in the gaps. So we identified the gaps and then we set out to fill them. We provided high quality, accurate testing and outstanding customer service. We provided online ordering and reporting. Believe it or not, in 2012, none of the companies in the United States could you order online. You could request a kit, but you actually couldn't pay money online and you couldn't actually tick a box indicating what tests you wanted. So it was all on paper. We offered the largest menu of tests and we really wanted to be one-stop shopping, and we currently still offer the largest menu of tests available. We continued to develop tests based on customer requests. So we would meet with our customers, we would go to dog shows, we would listen to our customers, and when a new mutation was identified in the medical literature, we would ask our customers, is this important to you? Is this something that we should develop? And we developed the only standards and guidelines for clinical testing in an unregulated industry. So when we started Paw Print Genetics, and in fact, it's still the same today, genetic testing in the veterinary space is unregulated. Well, of course, human testing is highly regulated, and this was the environment that we came from. And we set up our laboratory as if we were in a regulated industry. And then we published standards and guidelines, hoping that other laboratories would follow. So how are we different than our competitors? We reacted quickly to market indicators based on our interactions with our customers. We challenged the status quo by offering the largest menu of tests. We offered breed-specific panels of testing, which no one, of course, now everybody does because they copied us, but no one was offering breed-specific panels. So we made it easy for people to order. If they weren't sure what to order, they could just order the panel. And we had lower price points than some of our competitors, but scalable margins. We identified new markets to test, such as the designer breeds. We were actually the first company in the United States that would receive or accept a sample on a doodle or other designer breeds. The other companies wouldn't accept samples unless the dog was actually registered through the AKC. And of course, the AKC doesn't register designer breeds. We also started talking to hunters and went after hunting dogs and service dogs, such as guide dogs for the blind and other service organizations. We offered innovative tools to our customers, such as a color calculator so that they could predict the color of the puppies that their dam and sire would produce. We offer Paw Print Pedigrees, which is our social media site where customers for Paw Print Genetics can actually show off their dogs and show their results and contact each other. And we offered clear by parentage certification. So if the dogs were tested through us, we did disease testing in the parents, then the puppies could receive a clear by parentage certificate. Then we decided to differentiate and not just do dogs, but we launched testing for cats and for monomorphic birds. And by monomorphic birds, it's those species in which you can't tell the difference between the males and the females, such as the pigeon, for example. And so we offered testing so that you can sex birds and identify the males and the females. So this slide shows our product launches since inception. So again, in April 2013, we launched 86 tests, and we continued to produce and launch tests every year after that. The orange indicates our canine health check test, which is an all-in-one test. It used to have 150 tests when we launched in April, and in November 2019, we increased that to 250 tests. And then the purple indicates when we launched CAT scan, and green indicates when we launched Avian DX. And this shows just the product sales for the canine testing. And you can see in 2021, so in last year, we did more than 250,000 tests. So that's this number right here, in more than 65,000 dogs. So really incredible growth for a startup in Spokane, Washington, that didn't even know prior to 2012 that you could do genetic testing in dogs. We created customer loyalty through various programs, and you can see that in 2021, for example, the red indicates repeat customers and the blue indicates new customers. So we continue to get in new customers, but we have a very, very loyal customer base that continues to order from us. Well, this company wasn't without criticisms as well that we addressed. We went to our first Canine Genetics meeting, and from the stage, the person actually said that, oh, there's a new company out here, but they're human geneticists. What do they know about dogs? Well, first of all, DNA is DNA. Mammalian DNA is mammalian DNA, and eukaryotic DNA is eukaryotic DNA. So we knew how to deal with DNA. And in fact, the human genome and clinical syndromes are much more complex in humans than they are in dogs. And it was really interesting for us that most of the canine inherited diseases that we were testing were actually first described in humans. So we knew the human correlates to most of these diseases that we are testing. We were also told that no one will order panels. Well, if you look at the orders today, the majority of orders include at least one breed-specific panel. Panels were the way and are the way the human genetic testing is done. It's efficient. It's a way to quickly find out what the problems are so that they can be addressed. The other thing that we did was we went to breed clubs and organizations, and we actually allowed them to create their own panels so that their members would then use Paw Print Genetics. These are the various brands. So we diversified and offered products. Again, Paw Print Pedigrees is our social media site. Paw Print Genetics is our breed-specific testing. The Canine Health Check is our all-in-one 250 tests for your dog. The CAT scan is about 86 different tests for your cat. And of course, Avian DX, so that we can determine the genetic sex of a bird. We also published at Paw Print Genetics. So one of our first two papers was we published in two very rare breeds that had never been studied before. And we identified that they did harbor some genetic mutations. And so we published in the Dutch partridge dog, which is shown here on the left. And we published another paper in the Bohemian wired hair pointing Griffon, which is shown here on the right. We published the first international study of police working dogs. We collected samples from Israel, from researchers in Poland, and then the Smithsonian Institute, the Smithsonian Museum of Natural History, actually helped us collect police dogs in the Washington, D.C. area. And all of these samples were tested here in Spokane at Paw Print Genetics. And we published our first study looking at these working dogs to see if they harbored any genetic mutations that would shorten their careers. With the idea that if you're going to, police dogs start at least $10,000, a bomb sniffing dog by the time it's trained is $100,000. So we would hope that you would first do genetic testing on that dog to make sure that it's going to have a long career and that there isn't something that's going to be detrimental to that dog. Again, we published the first standard and guidelines for companion animal clinical genetic testing, and we published the fifth mutation found for long hair in cats, and this is found exclusively in the Maine Coon cat, which is shown here. That's the Maine Coon. And the final publication that I wanted to spend a little time on is that we actually found trisomy 38 in a dog. So the laboratory is now using microarrays. So we've come full circle from Signature Genomics using microarrays, and now Paw Print Genetics is using microarrays for certain tests. This is a picture of what the data looks like for normal chromosome 38. So dogs, remember, humans have 23 pairs of chromosomes. Well, dogs actually have 38 pairs of chromosomes. And this is a normal chromosome 38. And this is 2 copies of 38 because there's a bar right here that indicates 2 copies of this chromosome. This dog here, his name is Tippy, and he actually has 3 copies of chromosome 38. So he has trisomy 38, and we can see that because this band here splits, which indicates that there's 3 copies of chromosome 38. And instead of the bar being here at a normal 2 copies, it's up here at a 3 copy level. Tippy doesn't seem to care that he has trisomy 38. There doesn't seem to be anything, any clinical abnormalities associated with this. Shown over here is chromosome 38 in the dog, and it's made up of sections of chromosome 1 from human. So these are what's called the synthetic regions between chromosome 1 in humans and chromosome 38 in dogs. Humans that have duplication or triplication of these regions actually have medical problems. But Tippy not only doesn't seem to have any medical problems, he's also sired 2 litters. So he's perfectly happy having trisomy 38. And we find this intriguing and want to learn more about this dog and trisomy 38. This is the first aneuploidy, other than the sex chromosomes, the first aneuploidy ever identified in dogs. So how did we exploit opportunities to create disruptive change? Well, first of all, we use new technologies to replace existing methods. Microarrays completely replaced microscopes for cytogenetics in children with developmental disabilities. Both companies used high throughput technologies, but in Paw Print Genetics, it allowed for us to produce a low-cost breed-specific panels for testing. We improved diagnoses in both companies, and we published to establish credibility, which is very important. Both companies implemented new business models when we needed to. Signature had to pivot to sell signature chips and our software Genoglyphics in an already successful service laboratory to continue to compete with this competition that we were creating. Again, for Paw Print Genetics, we implemented breed-specific panels in canine testing. This had never been seen before. And now, of course, every canine genetic testing lab offers breed-specific panels. And we offered online ordering and reporting, which had not been seen in that industry. We used customer feedback to improve products or services, such as our Clear by Parentage program at Paw Print Genetics, or continuing to develop new breed-specific tests, and launching other tests. We often had our customers ask, well, why don't you test for cats? Well, now we do. And we expanded our customer base. Again, we sold signature chips. We licensed the Genoglyphic software. We offered testing for hunting dogs, doodles and other designer breeds, rare breeds that had never been studied, and service dogs. Paw Print Genetics is the world provider now for all service dogs in the world. So every service dog that's being produced through Guide Dogs for the Blind and other agencies such as that, the genetic testing before breeding is performed at Paw Print. And we developed other products such as the Canine Health Check to do a large scan of a dog if that's what a customer preferred, and CAT scan and Avian DX. And we created customer loyalty through our signature conferences, through our collaborative publications that we are published in the medical literature, through having online accounts with dogs and results. So our customers, we have many customers that have more than 200 dogs in their accounts because they're testing on their puppies, they're testing on their dams and sires, and everything is located in their online accounts. We created Paw Print pedigrees so that our customers could share results. We allowed clubs and organizations to design their own breed-specific panels, and we provide membership discounts to certain breed clubs. So I'd be happy to answer any questions, but before we go to the questions, I just want to thank you all for your attention. And again, I want to thank Todd for inviting me to share my journey. Thank you.
Thank you, Lisa. I unmuted everybody, so you should be able to ask your questions directly. There are a couple questions on the chat. I'll take a look at those to start. What changes, if any, did you see during the pandemic? Did you increase demand? Was there an increase in the need for adoption or fostering? And how did that affect your business?
So I often referred to this as our COVID silver linings. So there were some silver linings during COVID, but you certainly had to look pretty hard for them. This was a very scary time. In March of 2020, we were trying to figure out if we were going to be able to stay open or if we were going to have to shutter the business. And the employees at Paw Print were amazing. Anybody who could work from home worked from home. And then in the laboratory, which of course you couldn't work from home, we went to shift work. For a while, we had 3 different shifts, including the weekend, so that people could social distance, but we stayed open and we had a banner year in 2020. I talked to customers who were coming out of retirement to produce more puppies because so many people were staying home, everybody wanted a puppy. And so we had an incredible year in 2020. We had another incredible year in 2021. And so again, there were a lot of tragedies with COVID, but if you looked hard, you could find some silver linings. And certainly in the pet industry, we saw an uptick in our business.
Great. Does anybody else have any questions? I've got a couple more. Wonder if they can talk.
Can you hear me okay?
Yeah.
Yes.
I have a quick one. You'd mentioned earlier that you were willing to share some of your technology with some of your competitors, and it kind of, well, they weren't competitors at the time, but it created competition. Why was it so important that you felt like you had to do that?
So Signature was a very interesting company. It was extremely profitable and it grew very fast. And, it's important that a company makes money because you have to pay your employees, right? But we never set out to make money. In fact, I was convinced that we weren't going to, but it was something I felt compelled that I needed to do because I knew that when we were looking through a microscope that we were missing things. So when we started Signature Genomics, it was actually the first time in my life that I ever received a thank you note. And I received a thank you note from a family who had been searching for 10 years for a diagnosis. So it was very important to me, and I actually didn't even recognize at the time that I would be creating such competition. It was very important to me to share the knowledge, to teach others, because this was the way that cytogenetics needed to be done. And because we were in Washington State, we got a lot of samples from Washington because we were under Washington State's Medicaid. But let's say we weren't necessarily getting samples from Nebraska, for example. And so every laboratory needed to be able to do microarrays so that the children in their communities whose insurance prevented samples from being sent to us could get this testing done. So it just was the right thing to do.
Thank you.
Did that answer your question?
Yeah. The only thing I was thinking of was like maybe you could have considered licensing it, but I guess from an ethical perspective, what you did was probably the better option.
It's funny looking back on it now, it was absolutely the right thing to do. If you Google the history of Signature Genomics, you'll find that the acquirer shut it down in 2014. So only 4 years after purchasing Signature. So we left this legacy though, that every cytogenetics lab now in the world can do microarray testing. And that's what's important.
Thanks for the question, Liam. I've got a question through chat. How did you capture a baseline genetic line if there are so many types of dogs? That's the first part. In other words, did you employ a GWAS? I don't even know what that is.
Okay, so George is asking if we had to do GWAS. So GWAS is genome-wide association studies, perhaps using, sequencing. That's not what we did. So what Paw Print does is we go to the medical literature. If we haven't discovered the mutation ourselves, which we've only discovered the one long-haired mutation in Maine Coon cats, we would go to the medical literature, we would see things that had been published in dogs, and then we would develop the tests so that we could detect that genetic change. So even though we have a very robust R&D department, most of the testing that we do, the vast majority of the testing that Paw Print does, is based on information that we obtained from the public medical literature, and then we would develop the test.
Was that good enough for you, George?
I mean, we do employ GWAS and other technologies to identify new mutations, but that's not how we develop our diagnostic tests. And then as far as there's so many different types of dogs, we don't do breed identification. And so 99% of the dog genome, whether you're a Chihuahua or a Great Dane, your genome is the same. So we can do these tests on many different breeds of dog. We don't have to develop breed-specific testing. So I hope that answers your questions.
So we have another question here. I think this is from Matt Darcy Evans. What would you say was the key component which drove the rapid growth of Signature Genomics? And part two is, what important business ideas would you say are beneficial for the growth and profitability of tech-oriented startups?
So for your first part of the question, what drove the rapid growth was the fact that we went from making diagnoses in 3 or 4% of patients studied through a microscope to 20 to 25%. So we received samples from doctors, and doctors were getting diagnoses, and that's what drove it. That they were now getting answers that they needed so that they could, provide genetic counseling to the families, so that they could treat the children, so that we could publish these syndromes, so that others could learn about these disorders, so that they could learn whether or not it was associated with seizures or other medical problems to look out for. So there was just some early adopters, and then we published, and I think that's what drove the rapid growth, was just the fact that we were making diagnoses. As far as an important business idea for growth and profitability, I think if you go back to my first slide about the 5 ways to exploit opportunities, I think it's really important to understand your customers, because you don't want to produce something that no one wants or no one's going to buy, right? They have to want it. They have to need it. Hopefully they can't live without it, and they want to be able to buy it. And can you produce it at a price that they can afford? So that's the main thing, is to have, a great idea. I mean, I think there's a lot of great ideas out there, but it has to be something that people actually want to buy. Did that answer I hope that answers your question, Matthew. So then Brian asked about financing. So yeah. So with Signature Genomics, it was started with seed money, like I said, from Sacred Heart Medical Center and PAML. They were our business partners. They provided seed money. In 2007, we were growing so rapidly that we actually had venture capitalists coming and knocking on our door, which doesn't always happen. And so we actually did take on venture funding in 2007. And that was a very interesting experience. It's interesting because we went from having a scientific board run company to a VC-backed VC board. and the boardroom changed dramatically. We went from people who were concerned about making medical diagnoses in children and making sure that the technology was widespread, to a group of people who only cared about making money. So that was I have to say, it was very difficult for me dealing with a VC board. So for Paw Print Genetics, because I was able to, I actually funded Paw Print myself, and I did not take on external funding. So hopefully that answers your question, Brian.
And there's another question. You might have already answered it. How did you start Paw Print Genetics in terms of market research and financing? You
Right.
kind of already answered that, right?
Yeah. So for show me, to answer your question, the market research was simply using the other companies. I kept a notebook, which is actually still in my office at Paw Print, that outlined, what was it like to try to order the test? What was it like to try to get a kit? What did the results look like? And I just documented the whole process for all of the companies in the United States that I used with Trixie the Wiener dog. And then I just outlined all the gaps, and we simply set out to fill all the gaps. And apparently it worked, because Paw Print is actually the #1 companion animal testing company in the world. Now, there's other companies that do different things, like Mars Wisdom Panel. They do breed identification. There's another company that does breed identification. We don't do breed identification. So, but if you just think about making, identifying carrier dogs so that breeders can avoid producing puppies with genetic problems, Paw Print is, the leader in that.
Let's open it up to everybody to see if anybody has any questions here.
Lisa, this is Scott Rosick from 490. I'm curious if you have any particular insights as to why they shut the company down after 4 years, because it sounds like it ideally should still be in operation, but I'm just curious if there are any insights around that.
Well, so I stayed from 2010 until 2012, and no.
Okay.
Buy me a beer and I'll tell you, Scott. But yeah, no, it's, I think that, a couple of things with my journey. First of all, I've never read a business book. Now, I probably shouldn't say that to business students, but you can see I have a cabinet full of them behind me. People give me business books all the time. I've never read one of them. I've started most of them. I haven't read them all. Haven't read any of them cover to cover. I think that it was really important, at least for me, to not know that most businesses fail. And because I didn't think that Signature would fail. I didn't think Signature would be as profitable or as grow as fast as it did. But I didn't think that we would fail, because it was better. It was better than the current technology. It broke my heart that they shut down Signature, but in fact, most acquisitions fail. And it's sad. And, Paw Print was recently acquired, and I have very high hopes that Paw Print will be around for a very long time.
Would you mind if we unpacked the starting of Signature just a little bit more? Because it sounds orderly, but you're sort of giving up this salary into perpetuity. You're leaving being a professor. Camel and Sacred Heart fund you, but was there like a scary window where you just sort of moved here and you were raising capital, and if they hadn't written a check, you were going to be in trouble? Or was it as orderly as you kind of laid it out, the starting and funding of Signature?
so there were a lot of difficult times with Signature, but Signature was never scary like Paw Print was. The major difference was with Signature, I had such a conviction that this was a better way to do cytogenetics, and that and Skip Davis and Tom Tiffany were absolutely wonderful, supporting us in our venture, and supporting us in our vision, bringing on VCs in 2007. It got very rough with the VCs just because we had a different perspective. Mine was focused on the medicine and making diagnoses and improving the technology, going deeper and deeper in the genome, finding smaller and smaller alterations. And theirs was simply focused on making money. So it was frustrating. But there was never a day, because we were profitable in our first year of operation, and we were profitable ever since, every day after that. It wasn't like Paw Print, on the other hand, here I'm finding it myself with my resources. And we started in 2012 and we were not profitable. Our first profitable month was December of 2015. So Paw Print was very scary. And in many respects, Paw Print was much more difficult because Signature, we would do one microarray in a child for about $1,500, and insurance was covering it. And at Paw Print, our average price is about $80. So, we're talking, the scale is completely different. So that's why, at Paw Print, last year we did more than 65,000 dogs. I mean, we have to have this scale in order, to make money. And so, but after 2016, Paw Print really got accepted into this mature market. And we got customers such as the Assistance Dog International. So we did all assistance dogs in the world. We continue to have that contract. And, it got better. But I would say that Paw Print had many more scary moments. And COVID, 2020 was so difficult. It's hard to even express how difficult it was. With half my workforce, working from home, the other half having to be in separate shifts so that they're not, so that they're social distancing and not getting each other sick. And it just was really difficult.
So from 2012 to 2015, did you have in your mind sort of like an outer boundary of how much you were willing to put into the company? And did you go past that boundary through the scary times, or how did that piece work?
Yes, I had a boundary, and I ended up doubling it because I was so convinced that we were doing it better than everyone else. It's just that not everyone was hearing us or finding us. And it proved to be the right thing to do, but it's pretty scary when it's your own money. In 2000, I was still putting money in the company. So not every month was profitable. We had profitable years, but I was still putting money in the company even in 2018.
Sure.
So if we had to buy a large piece of equipment or something like that, but I just was convinced that we were doing it better than everyone else.
So what were you looking for was the second acquisition, was it unsolicited? Somebody just came and found you, or was there like a new approach that you took to try to find a better home for the second company?
So with Signature, it was quite interesting because this was in 2009, and some of you who are old enough to know, 2009, there were basically no acquisitions happening. And we actually had 4 different companies trying to purchase us in 2009. And I thought that we were doing the right thing by selling to that particular acquirer. And so I learned from that experience that you can't predict the future, that if you have a crystal ball, it's probably broken, and that it's, you just do the best that you can. And so in this particular interest, for paw print, we had a lot, many suitors. And again, I think that I chose the right home for paw print.
Great.
I have a couple other questions from some students and my own. Where do you see opportunities in the pet industry in the future? And any general advice for beginning entrepreneurship students?
So, I think the pet industry is huge, $100 billion a year industry. And I think that there's ways to get into the pet industry that you don't have, it doesn't have to necessarily be science, right? So in other words, Signature Genomics, we got samples from doctors and hospitals, genetics clinics, pediatric specialists. These people understood the information they were getting back. Paw Print Genetics is direct to consumer, to dog breeders and owners, and we produce complex genetic information. It's a difficult business. So I think direct to consumer is difficult. And then direct to consumer with something that's high tech, I think adds a layer of complexity. So I don't think I answered your question, Todd, but that's my answer. What was the second half?
Any general advice for beginning entrepreneurship students?
Oh, I have tons of advice. So first of all, set out to do something that you're passionate about, that you believe will make a difference. Never start a company thinking that you're going to make money. If you want to make money, then you can go out and make pet rocks. And I know that most of you are not old enough to know about pet rocks, but literally someone used to sell rocks and they were your pet rocks. But I don't think you should start a company with the idea that, oh, I'm going to make money. That is not a reason to start a company. You need to believe in what you're doing. You need to do something that makes a difference. And you need to really believe in your vision, because you will meet people that do not believe in your vision. I met lots of people who told me that Signature was not going to work. There were too many hurdles. By the way, I went out and licensed all the IP that I needed to license. You have to have the attitude that I'm going to get this done because it's the right thing to do and nothing is going to stand in my way. With Paw Print Genetics, we don't necessarily have the time to go into it, but we were sued 6 times, too many times to count. 2 of them ended up in federal court. We won both lawsuits. They cost me hundreds of thousands of dollars, and I would have never backed down because we were right. And in the 2 cases where we ended up in federal court, the judges agreed we were correct. We were right. So if you believe in yourself and you believe in your convictions, you have to have a strong stomach and you just keep moving forward. And that's my advice. If you want to be an entrepreneur, you have to have thick skin, a strong stomach, listen to the people that matter. Don't listen to the naysayers. Well, you can kind of bend an ear because sometimes they have some wisdom in there. But again, when you hear criticisms, turn them into opportunities. I hope that you understood that was the point of my talk today. Take the criticisms and turn them into opportunities for your business.
Thank you for that. I have a couple other questions. If anybody else has questions, go ahead. But I have one related to women and entrepreneurship. I know we've talked about this before. Is there any specific advice you would give to women who want to be an entrepreneur?
Do it. I don't really, like to think of, that I had to do things differently because I was a woman. I know that there were certain aspects that were more difficult because I was a woman. And I know that when you look at the statistics, it's something like, less than 10% of all CEOs are women, which are terrible statistics, and we need to change that. I think hopefully it's getting better and it's getting easier. And if you surround yourself with smart people and the right people that support you, you should be able to do this whether you're a man or a woman. I don't think being an entrepreneur is an easy thing to do. I only have one experiment, me. I don't know if it was easier or harder because I was a woman. I don't know. I just know that it's hard and you have to stick with it. It's really, really worth doing. It's really, really worth being your own boss and to have a vision and to create something and to see that vision through. It's just really worth it.
Lisa, we have some D2C entrepreneurs in the room. Could you add any extra color on what you found challenging about D2C? Was it that you self-funded? Was it capital oriented? Was it influence oriented? But I'm just curious to hear what some of those challenges were beyond the fact that it was a kind of a scientific and geeky product, but just to be curious to hear.
Well, for Signature, I think the biggest challenge in the beginning was that, I had been an academic and published a lot and had friends all over the globe. And all of a sudden I found myself very alone because my friends that were cytogeneticists felt very threatened. They felt that they were going to lose their jobs because they didn't know how to do microarrays and they didn't understand microarrays. And that for me, with Signature was probably the hardest thing was, just feeling, very alone, and that people that you thought were your friends now felt threatened by you. With Paw Print Genetics, I said that I would never take on, venture capital or other, investment interests. But I have to say now, in December, I went through the transaction with Paw Print. And again, I felt very alone. It was just me. I had advisors and I had attorneys, but my advisors and my attorneys, I pay them, right? So they didn't have the right kind of vested interests in the outcome. They wanted an outcome. We wanted to see the transaction go through, but sometimes I felt like they were willing to give on some things that I didn't want to give because they just wanted to get the transaction done. And so if you have investors, I would say the one positive thing now looking back is that they're vested in an outcome, the same outcome, hopefully, that you want. And they're sitting with you side by side through something as difficult as a transaction, because I don't think there's ever been a transaction on earth that was easy. There's always points of contention. There's always things that need to be negotiated. And it's nice to have someone at your side through that you're not paying like an attorney, right? So, I don't want people to walk away saying, oh, I'm going to figure out how to bootstrap this myself. I'm never going to take investors because, all they care about is money. There are some positive aspects, with investors, you're getting their knowledge. They should be experienced, and they can help you with things like, when you finally go through an acquisition, an exit.
I think you described entrepreneurship in general, though. Like that's, Elon Musk calls it staring into the abyss and chewing on glass. Like there's just an aspect to it where you are alone. Like even if you have your executive team, even if you have your great advisors, and there's a whole bunch of like scary moments that no one knows about except for you.
Yep. Yep. Me and my steering wheel. By the time I would get home, I would be in a much better space. But yeah. Right. Yeah. I have a lot of really loud rock music in my car.
Nice. Yeah. And then people will also say that you were lucky after all those chewing on glass moments and all the challenging things you had to get through to build the company, right? It's an interesting conundrum, but what an awesome journey you've had twice now too.
Thank you. Thank you. I think there is some involved, but I also, it's hard work and perseverance. There is certainly some luck as well.
We're lucky to have you as part of our community.
Thank you. Thank you.
Charlotte Spokane had a question for you. Does age matter in starting your business?
Yes. I'm tired. I had a lot more. I started Signature. So I started at Baylor when I was 30. My first job when I was 30 after getting my PhD. I was actually 29, but whatever, 30. And then I started Signature when I was 40. I started Paw Print when I was 50. And people keep asking me, what are you going to do next? And you know what, next, I don't know, but I do think it gets harder with age. I do. I hate to admit that. I just admitted that to like 100 people, but I think it does get harder. I think I just look back and I was just super quick, super smart when I was 40, and I was pretty smart and pretty quick when I was 50, and I just don't know now. So it may not be the answer you wanted to hear, but.
Is that good enough, Charlotte? Wherever you are?
Yeah, I thought that was pretty good. I guess I was kind of wondering, like, maybe if you're like too young, will people maybe not take you seriously, or how do you know about that sort of stuff also?
Oh, I think Charlotte, that's a definite problem out there. Being a young woman, I think that's probably 2 strikes against you, right? But you just have to be convincing, and you can be convincing about your vision because you have a conviction, you believe in what you are doing, and you believe that it matters, and you'll build a story. So, be sure and have, advisors that you trust that can help you build that story. Your, what's called the pitch deck. Some are long, some can be short, but they need to tell your story, and people need to walk away going, yeah, I get it. I see Charlotte's vision. So, when I was just out of graduate school, even though I wasn't starting a business, I still had research, and I had to get grants, and I had to convince people that my research was important so that they would fund me. And, I think that if you can be an entrepreneur in lots of different ways. And I think even in doing research and getting grant funding is being an entrepreneur, because you still have to convince people, right, that to see your vision and that they need to buy into it. So it is harder when you're younger, but with, with the right advisors and with the right vision, I think that anything is possible.
What's the best and the worst part about being an entrepreneur?
The best is being your own boss. And the worst is being the boss. I mean, the buck stops with you. So you have to be willing to make those hard decisions. And that's the hardest part, is hopefully you're not making them every day, but I had to make some really, really difficult decisions. And even deciding, whether or not to sell the company is a really, really difficult decision. I mean, I could have kept running Paw Print and I think it would have done fine. I just felt it needed to go to the next level. And I think I'm really good at starting stuff. And I think I'm really good at having an initial vision and putting groups of really smart people together. But taking it to that next level, I kind of learned with Signature. Signature got to be 120 people. That's a lot of people to be the boss of. a lot of infrastructure that you need to have in place. And Paw Print was getting to that point where it needs to grow. And so I really felt it needed to be someone else. So I think that's the hardest part is being, the buck stops with you, making those hard decisions. But I love being my own boss. I thought I was a pretty good boss. I think I was a terrible employee, which is why I didn't last long after the Signature acquisition. I think I was meant to run the show, and so I'm just not a good employee.
That's pretty typical for entrepreneurs.
Yeah, so I've heard.
I'll open it up to the guest if you have any other questions. I have another one, but I'll let you guys go before me. Scott, do you have another one?
Lisa, you already stole the question, what comes next, but it sounds like, are you contemplating starting another company, or are you thinking advising, investing? Is there a path that feels better out there for what comes next?
So I've actually already started another company. Probably doesn't surprise you. But so I actually invented something. I've actually invented something. And so I guess what I want people to know is it's really possible to invent something that doesn't exist, because I have searched and it doesn't exist. And now I need to decide if I want to commercialize it or not. And I haven't decided. So, and I really don't even think I want to, and then here's the thing is, do I want to commercialize it myself, or do I want to just license it to someone else? And in both of those scenarios take a lot of work. So I haven't decided, but the good news is there still are things out there that need to be invented. And as far as investing, it would just depend on if the right opportunity came along.
I have a question on negotiations. I'm not sure if I should ask it, but I'm going to. What advice can you give us regarding negotiations with venture capitalists, or just negotiating in general? Because that's a really important part of being an entrepreneur.
I think it's just really important to have high integrity. If you're going to negotiate something, you have to believe that you're being fair, because you will be convinced that the other side is never being fair, or at least I always was. Negotiations are hard. And you just have to make sure that you have very high integrity, that, during a negotiation, you typically have to disclose things, that you're being, open and honest. And I've always been a good negotiator. I don't know why, but just like when I, with Signature, what my chairman had told me, no, you can't start using this because there's these IP, intellectual property that's out there that you would have to, license. I just figured out how to go talk to, Affymetrix to license it, or Roche and go and license it. I mean, you just figure out how to get it done. And I was able to negotiate. We needed 2 pieces of IP we need to license at Signature, and I licensed both of them for really good terms. And so again, I just don't think you can be too honest. And I don't think you can be too transparent. I mean, yes, I've walked away from some conversations kind of kicking myself going, boy, should you have really said that? But I think that in the end, integrity wins. So
Reputation.
And reputation, yeah.
I don't have anything else. Anybody else have a question? It's about 7:30. Molly, got a question? If not, I want to thank Lisa for coming in this evening. She's done a great job. And we're very fortunate, again, to have her in the community. And she's also part of the advisory board for the Hogan program.
For the
And we want
NBL.
to thank you for doing that. You've been on that since, what, about 10 years now, I think.
It has been 10 years with the NBL. Uh-huh.
Yeah, thank you for that. And I'm sure if anybody has any questions, she'd be more than happy to answer them. And I can send out her contact info if you want to contact her. And we're going to send you a nice gift out for doing this for us this evening, Lisa. Thank you very much.
Todd, there was one last question there on in the chat about Lisa, if you would be open to being linked on LinkedIn with the students.
Oh,
I'd love to
yeah.
know if that's a great strategy.
Yeah, absolutely. You can find me on LinkedIn. I also, it looks like I didn't, Carrie DeBoer asked if there were ever a moment that I wanted to quit. And I'll just quickly say, yes, lots. And you just deep breaths and really a lot of good rock and roll in a very loud car. Yeah. That's what gets me through it.
That's Scott's strategy too.
Yeah. So, well, thank you, everybody. I enjoyed this very much.